I'm Lowri Wigley, and my summer studentship was on investigating the role of helitrons in the emergence of novel Fusarium oxysporum lactucae isolates. I carried out my research project at NIAB, the National Institute of Agricultural Botany, though my actual work was all done remotely. It promised to involve bioinformatics, which combined biology, computer science, and statistics, giving me skills for use in a biological context. This appealed to me as I had already volunteered in a lab this summer and wanted to explore the different types of career made possible through my degree and my interest in plant-related science. Fusarium oxysporum is a fungal species complex including pathogenic and non-pathogenic strains. They are grouped based on host specificity, with, for example, lettuce infecting strains being known as phola. The wilt caused by Fusarium is becoming a worldwide threat, causing crop losses of up to 50%. The emergence of a new, more virulent race only enhances this threat, and therefore more understanding of the fungus is needed to help prevent further yield loss in the current environmental climate. To understand whether helitrons, a type of transposable element, had a role in the emergence of a newly discovered race, we used bioinformatics. This involved coding, which I'd never been exposed to before, and took some time to learn. I used a remote connection to the NIAB HPC to carry out all my analysis through installing a Linux system and using VS Code. This image displays one of the genomes we were working with in the VS Code software. We used an application called Genius to visualize the genomes and later genes of interest. We used different coding languages like Bash, R, and Python to run different analyses of the genomes, discovering helitron predictions from 3 different tools, helitron density on each contiguous region or chromosome, and the number of helitrons on core or accessory chromosomal regions. We found that the newer race had more on the core regions, perhaps explaining its virulence. We used functional annotation tools to identify if there was anything of interest for the resistance of the races, finding caseins and resistant genes, particularly in the newer genome. This will allow for honing in on these genes in later research. Our final figure was a heat map that showed the relationship between the races and how this may have evolved from each other based on their helitron region genes. This placement allowed me to enhance my scientific knowledge through teaching me how to code, a crucial skill in this day and age, as well as showing me that I actually enjoy it. Moreover, I was given the opportunity to give a presentation and also to critically think about the work that I was carrying out, how to move forward. Moreover, I learned about a career that I may have not considered before, showing me that plant science is incredibly varied.